Hello, students. Now, here we go that we have to understand the concept that the definition of biotechnology. So, when it goes to definition of biotechnology, students, the way to say is it is a kind of integration of a natural science and organisms, cells, parts thereof and molecular analogues for the products and services. That is how that a definition has been put by EFB. What is EFB? EFB meaning European Federation for Biotechnology. This is a one federation explaining the definition, integration of a natural science and organisms. Okay, the organism has been integrated, the cells, everything. What for? It has been integrated for to get a products and also for the services part. Now we can also understand in a very very simple term as now let's take the microorganism support, or let's take the organism support, and then we have to get the products from that particular microorganisms by growing it, and then that is in the laboratory, and by using the technology or by using the skills. Okay, now ultimately by those kind of products we have to get a profit, and this kind of a manipulation should be done in the lab by manipulating the genes. The genes has to be manipulated. So by this kind of processing and technology, we call it the subject called biotechnology. Now the next part of it, what we have to understand is principles of biotechnology. There are two principles in the biotechnology. Number one is genetic engineering. Genetic engineering. Number two is maintenance of sterile conditions. Now, what is maintenance of sterile condition? Means always, whenever we want to grow the microorganisms in the lab and we want to develop those kind of things, it should be in a pure culture. The pure culture, there should be no impure kind of a culture, there should be no contamination. The contamination free environment should be made. That is exactly where one of the major principles of biotechnology, and this is the one keenly has to be taken care. So otherwise the whole project will gonna collapse. So very very important is maintenance of sterile conditions. Okay, so let's come to the genetic engineering. This is an aspect where we need to understand. We need to understand that this genetic engineering also can be used in a different terms. That is, we also call it up the gene cloning. We also call it up RDT. RDT meaning recombinant DNA technology. These are the different kinds of words that we use it. Now, let's also understand, after knowing what are the two principles of biotechnology, the second point is traditional hybridization. So, what is the difference between traditional hybridization and the modern hybridization? The traditional hybridization, students, if we want to breed, then for the next generation, both desirable genes and as well as undesirable genes. Okay, desirable genes and undesirable genes both will be transferred. We are cannot able to avoid the undesirable genes in the traditional hybridization technique. Whereas what we took in the modern hybridization technique, here only desirable genes are transferred. So that is what the technology is. That means completely that whatever the undesirable genes can be avoided by the modern hybridization. That is a technique of it. Okay. Now, next, now this is nothing but what we are talking is about the gene cloning steps. Gene cloning steps involved, first we have to select the desired gene. Okay, the desired gene or the foreign DNA has to be taken at whichever the from the host or the kind of interest that we have to isolate. So that is a desired gene, the interested gene that has to be taken, and then it has to be inserted into a vector. Vector is nothing but a vehicle which carries the desired gene. Vector is a vehicle which carries a desired gene. The vector could be plasmid, could be plasmid. Now that vector has to be inserted into the host. Now the vector, if it is with the desired gene, we call it a recombinant vector. Now that has to be introduced into the host. Look at that. The host that we are selecting, the basically we select the host which is a prokaryotic host. There are two types of host: prokaryotic host and eukaryotic host. Generally, we select prokaryotic host because it is of a faster multiplication. That is, the multiplication will be by the binary fission. The two daughter cells will be produced. So the doubling time is very, very less. So hence, we go for such kind of aspects of understanding. But here, students, that you have to understand that vector what we have selected is called as a plasmid. Generally, we select plasmid only. The plasmid, why plasmid? Plasmid is nothing but it is a small double-stranded DNA, which is has to be called as extra chromosomal DNA, and this is having a property called self-replicating. Look at that word. Self-replicating is one of the fantastic kind of other word, that is gene, that a characteristic the plasmid has. Now, this is overall what we are understanding is about gene cloning steps. Now, coming to the little history of a kind of the subject in the case of a genetic engineering. The first point here is there were two scientists. The scientist name is Cohen and the Boyer. In the year 1972, what they did was they transferred a gene from Salmonella to the E. coli. Look at that. They transferred from Salmonella to the E. coli. What is a gene this transferred is antibiotic resistance gene. Antibiotic resistance gene was selected. Okay, so what is antibiotic resistance gene means? Suppose a E. coli or the salmonella, which can able to grow in the antibiotics, it can able to withstand. There will be no effect from the antibiotics. The salmonella will not be killed, or the E. coli will not be killed because of the antibiotics. So that resistivity will be developed in that case of a bacteria. So that gene we have to transfer. We can transfer from one bacteria to another bacteria. So that is the first kind of experiment that did very successfully in the year 1972. Next, number two things, the second point what we are understanding is the person by name, his name is Harbour, ARBR, who who also made a kind of experimentation that suppose a foreign, the infectious thing which is entering into the bacteria. Let's understand a virus. The virus which is entering into the bacteria. Okay. Now that virus can be called as a bacteriophage. Now, once it enters a bacteriophage, what is the bacteria will do now for to restrict the kind of the foreign agent or protect from the foreign agent? The two things happened that was observed by Harbour. Now, what he says inside the bacteria there was an enzyme which has been the enzyme. This enzyme name is called methylases. The methylases has made some modification in the host. In the nucleus region, it made some modification to protect from the foreign agent. Now, That is where exactly we need to understand the modification, that is for the protection sake it has made a modify. Now what is exactly by, unfortunately he also observed that, Arbor observed that, so there was another enzyme in the nucleus that produced, and then this enzyme name is called restriction endonucleases. Restriction endonucleases, and this restriction endonucleases, what it did, it was cutting its own DNA. It cuts its own DNA, and some kind of restriction was been made. So that was a very good, a beautiful kind of identification was done by the Harbor. Okay, so then he proposed that restriction. There is something called as that its own DNA has been cut. So it is very interesting kind of a thing. Then second thing he was telling, the methylase is that does modified. How it has been made modified? By adding the methyl group. Adding the methyl group. That is how it has been made. Number third concept, that is a Smith. The Smith, you know what he has to understand that this he made observation of all these Arbor, whatever the Arbor explained it, taken all these particular concepts, and then he discovered one of the restriction endonucleases. That name is called hind 2, hind 3, hind, sorry, yeah, hind 2. Hind 2 was introduced by the Smith. And yeah, what is hind 2 meaning? Hemophilus influenza type D. Now that was introduced and discovered. That was the first restriction endonucleases. Please make a note of that. That was the first restriction endonucleases. The fourth point in a history based, we have to understand Eriki is a scientist, a person who termed or called the word biotechnology. And the fifth person will also say Van Berg. Van Berg is the first person who is to introduce, we do the kind of transferring the genes. Okay, so hence that is the person is been called as a father of biotechnology. Father of biotechnology. The next we have to understand what are the tools in the subject of biotechnology. The first one is DNA manipulating enzymes. The first tool is it's all about enzymes. And second tool is a vector. We were talking about the vector is a vehicle that carries the desired gene. The third one is a host. When it comes to the host, there are two hosts, prokaryotic and eukaryotic. The prokaryotic multiply faster, doubling time is very less. Hence, we usually select the prokaryotic, but the eukaryotics are slow. Fine. Now, when it comes to DNA manipulating enzymes, we have restriction endonucleases that has to be understood, and polymerase enzymes, ligases. These are the different varieties of enzymes has to be understood in the case of the DNA manipulating enzymes. Now, let's go detailed study of it. The first point here, so we have taken a subject that is enzymes now. The enzymes in that case, the first thing that we need to understand is about the nucleases. The nucleases meaning it, they are the group of enzymes that cuts the DNA. Look at that. So suppose now this is the double stranded DNA that has to be cut. That has to be cut. Now that is the enzymes called the nucleases. Under that, there are two different kinds of nucleases. One is called exonucleases, another one is called endonucleases. Exonucleases will gonna cut. It will gonna cut at the end. So now understand, now this is the double stranded DNA at the end region. So at the end region, it will gonna cut. Whereas another enzyme that's called as endonucleases. Endonucleases, it will gonna cut internally. Internally, it will gonna cut the phosphodiester bonds. So what is phosphodiester bonds? Already you know it, the kind of a basics where the bond which is there between the nucleotides. Right? Now, internally cutting is called endonucleases. At the end cutting is called as exonucleases. Now we need to know another important, most important to understand our subject, that is palindromic sequences. So what are palindromic sequences? Palindromic sequences meaning, that means there is a repeated sequences are seen. There are repeated sequences are seen, and that sequence length will be 48 nucleotides. The what is repeated sequences? When we read in the forward direction also, the same thing when we read from the backward also, the same repeated sequences are seen. Now that is what is called as a palindromic sequences. Now, for example, when we read from the forward like this, when we read the forward, GAAATTC, I have read in a forward direction, in a forward direction. Now, when in the same way, we need the same repeated things will be, we have to understand here. Okay, so look at that GAATTC. Again, I'm reading the same thing, GAATTC. Like even when I read forward also, when I read backward also, the same sequences are repeated. Now that is what we call it up as a palindromic sequences, and that length will be we are telling is 4 to 8 nucleotides. The length will be a 4 to 8 nucleotides. That's a speciality here. Now let's understand restriction endonucleases. Now we are coming to the actual point of the subject now, genetic engineering, that is restriction endonucleases. Now what is expansion variant restriction endonucleases? Which was the first restriction endonucleases discovered? That was Hind 2, that was by Smith. Till now, more than 900 restriction endonucleases are discovered. They are only discovered, only can be extracted, only can be isolated from the prokaryotes. Look at that, speciality, only for by the prokaryotes. And let's understand the restriction endonucleases nomenclature. Means we after we discovered, it has to be named, right? In that case of naming names, for let me take an example, that is eco R1. Eco R1 is one of the famous restriction endonucleases. So whenever we write the name of the RN, we will be writing in an italic form. Look at that. We have to write it in italic form like this: eco R1, italic. In italic form, we need to write. Now, when we write eco R1, so E stands for, that is the first alphabet. It should be written always in a capital. Yeah, that E stands for is the genus name. That E stands for the genus name here. The E stands for Escherichia. Escherichia, that is the E. And when it comes to the next two alphabets, look at that next two alphabets. So CO. CO means that is coli. Coli, which is a species name, has been put. The first two alphabets of the species has been taken, so that they have written it as eco. Now coming to the next alphabet, look at that, R has been written over there. R stands for, actually, to meaning that R stands for rough. R stands for rough strain. Okay, so that means here we need to understand the rough strain and the smooth strain. It depends on the nature of the strain. That has been written as a R. Now, coming to the Roman letter 1. So, this is the Roman letter 1 understands that the order of the sequence. So, when it is written 1, that means the first order. If it is written 2, then it is the second order. It's all about order of discovery, what we have to understand in that particular strains, right? So particular strains. Now this is the one, how exactly this has to be understood. The nomenclature, the naming of the restriction endonucleases. Now this eco R1, what we are telling it about the restriction endonuclease, this has been isolated by the bacteria, that is E. coli. We called up that particular strain as, look at that, eco RY 13. So eco RY 13 is a one. From that only we have been extracted this restriction endonucleases, that is eco R1. Okay, fine. Now that is where exactly we need to understand the concept of it, which has to be understood, that restriction endonucleases. Now Little further, we'll go detail. Restriction nucleases are of three types. One is type 1, type 2, type 3. Type 1, uh, when it comes to type 1, type 2, type 3, type 2 is the one we are more interested in. Why you are more interested in type 2? Only because it is involved in the genetic engineering. It cuts at specific sites, and it is very easy to use, easy to cut, easy to demonstrate. Now this is where exactly, and of course our concept is genetic engineering, hence we need to go for it. Now, working principles of the restriction nucleases, look at that. The restriction nucleases, we already said that it will gonna cut at specific site, correct? Now we have to understand it will cut, little away from the center, little away from the center of the palindromic sequences. What is palindromic sequences? There will be repeated sequences, isn't it? But little away from the center, where when it cuts little away from the center, the opposite sequences, only one nucleotide sequences will be there. Okay, now that is out. Now for example, let's understand that if we are taking the eco R1 as an example for restriction into nucleases, and we are working on it, look at that particular sequences. This is a sequence of eco R1. Yes, GAATTC 5 prime. Now the other end we have to write it as 3 prime. So it has to be in the opposite sequence, should be 3 prime to 5 prime. The GAATTC, and the other prime is like this. Okay, GAATTC. Now what they are understanding is the restriction nucleuses will gonna cut little away from the center. See, here it is cutting, it is cutting this side, and similarly it is cutting here. So little away from the center, when it cuts, the opposite sequences will be only one nucleotide. So that is a speciality of the restriction in nucleus cutting. But when it cuts like this, when it cuts like this, it will gonna give sticky ends. Look at that word, sticky ends. Okay, or staggered end, staggered, which is sharp, cohesive ends. Okay, that is where, so one of the cutting the principles of restriction endonucleases. Example already we written, one of the example, that is eco R1, BAM H1, hind 3, PST1, SAL1. These are the few specific examples of giving sticky ends, which gives us sticky ends only. Now another kind of the working, where we have to understand the concept like this will gonna give blunt ends, but there it used to give the sticky ends, but this will gonna give the blunt ends. If that is the case, which one you will gonna prefer for the genetic engineering working principle? So obviously it has to be where, that is, sticky ends has to be taken for the process, because it is very easy to for the further for the joining purpose. So hence, okay, so look at that, blunt ends are also called as the flushy ends, flush ends. Now this is a sequence we can see. That's the sequence of SMA, this one SMA1, CCCGGG. Similarly you see CCCGGG when we read forward and when we read backward. Now there is a cutting. Once there is cutting, what happens? So it cuts at the center exactly. Now this will become one piece and this will become another piece. Now there are two pieces have been formed, but these two pieces are the blunt ends. So it is not easy for the next process. Suppose if you want to use the same kind of sequences, meaning again we have to modify that into sticky, then only it is possible to deal things. Now look at that, there are blunt ends, there are different varieties of examples. ALU1, SMA1, EcoR2, Hind2, SCAD1, E3. These are the few examples of blunt ends. The next two students. Now, basically, you have studied in molecular biology also what is the function of DNA polymerases. Polymerases meaning that is a polymerization of the DNA is done, where exactly the nucleotides has been joined by using the DNA polymerase 1, 2, 3, and all you would have studied in molecular biology concept. The same here also. DNA ligase. So DNA ligase, it is also can be called as a molecular glues, whereas restriction endonucleases are called as molecular scissors. Right? What is the function of DNA ligase? DNA ligase function is to join the phosphodiester bonds of the two pieces of nucleotides. Okay, the joining of the phosphodiester of the nucleotides of the two pieces of, okay, so sequences. Now that is the function of DNA ligase. Ligation meaning join. Ligation meaning to join. So other than this, we also have another enzyme that's called alkaline phosphatase. This is the one which removes a phosphate. Please make a note of this. Is an additionally we need to focus on that. So it's all about the enzymes what we talked about. Okay, so the tools of biotechnology, tools of genetic engineering. The first one is the enzymes that we studied. The first, that is DNA manipulating enzymes, we studied already. We have already understood the host, but now we will understand the vectors. So the vector is the one that we'll understand now. So in that, like yes, the essential features of the cloning vectors. The cloning vectors should have certain properties, certain characteristics are very important. The first property, that is origin of replication. Origin of replication is one of the major kind of property it is needed here. Why? Because suppose a foreign gene is called alien gene. You can call it an alien gene also. Once we introduced into the vector, for example, take an example of a plasmid. The alien gene has to fit, has to join, or has to be placed at a site that initiates the replication. Suppose somewhere you are dropping means it will not initiate for the replication process. You have to drop a person to the concerned destination only. So then only he will get into his house. Somewhere you are dropping somewhere 1 kilometer away, 1 kilometer before to his destination. That is not the way of dealing the things. You have to drop. You have to drop to the correct destination. So then only it is possible that the initiation of the replication will gonna occur. The second point is, I hope so now it's understand what is exactly ORI means. ORI means origin of replication. This is the one which initiates for the replication process. Clear. Number two, selectable marker. What is selectable marker? Selectable marker is a one which can able to remove the non-transformants, something not required things, unwanted things, but allows the desirable things. Look at that. The undesirable things can be removed and the desirable things can be added over there. The allowing the transformants, removing the non-transformants. For example, the transformants in the sense we want to introduce the genes like ampicillin resistance gene, tetracycline resistance gene, lag Z gene. These are all come under as an example of selectable markers. Are you ready? Now, selectable markers. These are selectable markers. Means ampicillin, tetracycline, lag Z should be there inside already. Now, look at that. Now, when we draw the plasmid, now here there is tetracycline. Understand there is a tetracycline. Tetracycline is a what? Selectable marker. Now, what we are doing is now we are introducing the desired gene now. Look at that. This place we are introducing the desired gene. Desired gene is introduced. Where we are introducing the desired gene? Into the area where exactly the tetracycline is already present. Now, if already present in that area when we deal, So that means that tetracycline, whatever earlier present, will be removed. And what will be added? That is desired gene will be replaced and added. So once we replace and added, so then the desired gene will introduce over there and the starts, replication or whatever the next process. No, now what guarantee that desired gene has been replaced over there? So for that, because it loses its tetracycline resistivity there, that is why we can able to guarantee that desired gene has been added. So hence that is the reason we are using the word called selectable marker. So otherwise there is no guarantee that yeah, desired gene are already transferred. It will be a successful story we cannot able to understand. So that's the reason that we are cross checking it by using these selectable markers. So I hope so. Now this is the second point of the essential features of the cloning factors. Now the third point is a cloning sites. Cloning sites means where that whatever the desired gene has been put, inside, so it has to develop into clone or the multiplied kind of a replicated kind of a replica or the copy, whatever the exact copy we can call it, that has to be produced. That is the region where exactly it has been produced. That is called as a cloning site or recognition site. Also, we can call it up. Is that now the question is that only one gene will be introduced into that particular vector? Can we carry only one gene? No, it's not like that. We can carry many genes. So because there will be multiple cloning sites, look at MCS will also be there, or we call it a polylinker. There will be single cloning site. Basically, single cloning site will be easy procedure, whereas a multiple cloning site is also, it is also can be carried, but little complication, little procedure will become a little hard. That is the thing MCS means. What is ROP? ROP is a kind of that region where replication of the plasmid occurs. Right now, these are the three different features that we need to understand in the case of a cloning vectors. Now, coming to the plasmids now. Yes. Now these plasmids, what we are discussing, these are the recombinant plasmid. That means already we modified in the lab. So we constructed in this in the lab. So that's what we are understanding. Recombinant is the title. We have to put it as a recombinant plasmids. Yes. The first one is a PBR 322. We are learning the PBR 320. In that case, 322 is what is 322? 322 is the strain number. The P stands for is a plasmid. BR stands for Bolivar and Rodriguez. These are the two scientists who introduced this, constructed this particular plasmid in the lab. Bolivar and Rodriguez. And its size is a 4.3 kp, kilobase pairs, right? 50 to 100. See, look at that. Once the desired gene has been introduced to these, 50 to 100 copies can be produced. Many, isn't it? Okay, so that is where exactly PBR 320 explanation. Look at this right hand side, the diagram. So already we have two selectable markers. Tetracycline resistance towards the right side, towards the left side we have amphicycline resistance. Look at that, everything. This is the ori site. That's the first important essential features that we said. Origin of replication, and these are the two selectable markers. So we can put many different genes inside. That is, multiple cloning sites are there. How you are identifying multiple cloning sites? So we can see that on the right hand side many restriction endonucleases are there. Look at that. What is the function of restriction endo? To cut, and ligases will be there to join. Okay, so this is how we are identifying it. Now in this PBR 322, to construct this, to construct the PBR 322 plasmid, so we are using the two selectable markers. So that means twice we have to plate it, twice or many times we have to plate it in the lab to get to color, to construct this, and to do the procedure of selectable markers. So it's a kind the time killing procedure, the cumbersome procedure by using this PBR 322 process. And now, because of that, that PUC8 has been introduced. PUC8 is also one of the next developed kind of plasmid, recombinant plasmid. Again, P stands for is a plasmid, UC stands for University of California. Okay, and this is a one, this is a which can able to produce more number of copies. It is also smaller, 2.7 kb, 500 copies can be produced. Look at that, extraordinary kind. This in this PUC8, there was a selectable marker. The selectable marker is called LAGZ. LAGZ selectable marker was there. LAGZ is a gene. That's a gene which will produce. It can be also called as a chromogenic substrate. It is LAGZ is a chromogenic substrate. Chromo means colored, which can give the colored reaction. Okay. Now, for example, now what color it is giving? Blue color it is giving. Blue color it is giving. Now, for example, now the LAGZ gene is there. Now look at that. This is the plasmid. Now here there is a LAGZ. So what is the function of LAGZ gene? So this will gonna produce an enzyme. This gene will gonna produce beta galactosidase. It's an enzyme. Now this, what is the function of this enzyme? This will gonna convert the lactose into glucose plus galactose, right? Glucose plus galactose. This reaction will be done because of the enzyme, and it gives what colored product? Blue colored product. Blue colored colonies will be given. Now, what is the intention? Now the desired gene has been introduced to this area. Which area? LAGZ area. Then LAGZ gene will be inactivated. Look at that. That is the process called insertional inactivation. Clear? Insertional inactivation, meaning desired gene has been inserted, but the LAGZ gene is inactivated. If it is inactivated, it will not gonna give rise to blue colored colonies. It gives colorless colonies. It gives colorless colonies. Now the next point that we need to understand, difference between plasmid DNA and as well as a chromosomal DNA. The plasmid, what is plasmid? Plasmid will have it is seen in the bacteria prokaryotes as per the bacteria concerned. So there is no histones over there. It's a circular DNA, small DNA, self-replicating. There are no vital genes. Whereas a chromosomal DNA, which is there in the eukaryotic, which is a linear shaped, there are histones and also the vital genes. That's a kind of differentiation that we can see between plasmid and as well as a chromosomal DNA. Now, coming to the bacteriophage, can also act as a vector here. Basically, when we take a chapter called vector, the first point that we have understood is the recombinant plasmid vector. That is PBR 322 we studied. We studied PUC8, which has been constructed in the lab. So three different features also we studied over there. Now the next vector that we are talking is about a bacteriophage. What is bacteriophage? This is a virus actually. Now, but what is the comparatives between the plasmid, the recombinant plasmid, and also bacteriophage vector? Now, this is the one which gives highest copy number. Please make a note of this question will be asked. For example, M13 is a one which can be used. Cosmid, what is cosmid? Cosmid is also a vector where how it has been prepared. It is prepared like cohesive sites plus phage. Okay, cohesive sites of the plasmid plus phage. YAC is considered to be one of the largest the vector expansion of the YAC East artificial chromosomes, we call it up. What's the speciality? Now here there are certain genes called telomerase genes. Telomerase genes will be, that's a speciality here, right? Largest vector. BAC, bacterial artificial chromosomes, and it has got certain speciality, that is fertility genes will be present inside. Phage mid, that is phage plus bacteriophage plus plasmid, that is why the name is phage mid. Transposons are called as the jumping genes. These are also considered to be the vector which can able to carry the genes. Okay, so again it has got its own speciality of it, which is can jump. TI plasmid, tumor inducing plasmid, it's called as, if we modify that, so it can able to decrease its pathogenicity will go off, and finally we can able to transfer the desired gene in it. Look at that, the bacteria name is called Agrobacterium tumefaciens. Agrobacterium tumefaciens is a one the species which is related to this TI plasmid. Whatever the plasmid, it's called as TI plasmid. And TI stands for is a tumor inducing, and T stands for is a transfer. T stands for is transfer. Retroviruses can also be modified. Modified retroviruses can also be used as a vector. Now, these are the varieties of vectors we have, so which is used in the biotechnology, used in the genetic engineering concept. Okay, fine. Now another special kind of a vector also we have, that is called yeast episomal plasmid and modified TI plasmid, which can act as a shuttle vectors. So what is shuttle vector means? This can be used for both prokaryotic host and as well as eukaryotic host. Look at that. For both the host, we can able to deal. Now that's about the vector. The next process of we have to understand the processes and of genetic engineering. In that, the first process like this we need to understand, that is isolation of a DNA is the first process we need to. After that, we need to study gel electrophoresis. Then PCR technologies we need to study, and then we have to go for insertion of the DNA into the host, how it has to be. So these are all we need to understand in the particular concept of it. Okay, so now one by one, the steps of the principal process of RDT. RDT means recombinant DNA technology has to be understood. Okay, so we will continue, okay, in another session.